Hello, I'm Dave Bolton and welcome to Under His Righteousness. Today, let's take a look at the subjects of DNA, RNA and the genetic code and how they relate to the Bible. There are two forms of DNA. One is sited in the nucleus of the cell and the other is the power station of the cell. It took from 1869 to 1953 to discover all the components of DNA and that it consists of a double helix similar to a spiral staircase or ladder. The two spirals consist of sugar and phosphate, which are joined together with steps or rungs called codons. There are four types of codons, which are represented by the letters A, C, G and T. There are 64 possible combinations of these types of codons. The chemical makeup of the organic bases are each designed to combine only with another specific single base. For example, A can only link with T and vice versa. C can only link to G and vice versa. Bases A and T cannot link to C or G or vice versa. If the DNA molecule splits in half, every base combines with its matching opposite number from the nucleotides moving freely in the interior of the nucleus of the cell. Each half acts as a pattern for the copying of the other half. When the two DNA halves have replicated, the DNA molecule becomes two. This process is only possible with the transfer of proteins and enzymes from the cytoplasm, which is in the main body of the cell outside the nucleus. This transfer causes the nucleotides to be constructed in the correct order and fused together. This is done in two phases, both involving RNA. This amazing process is extremely complex, especially when you consider that a DNA molecule can comprise of literally hundreds of thousands of coils, which in turn contain millions of nucleotides. When duplication begins, the DNA molecule has to uncoil itself, and the millions of nucleotides have to be constructed in the correct order and fused together by enzymes from the cytoplasm of the cell. The speed at which this process takes can be truly amazing. If we take a bacterium as an example, it takes approximately 20 minutes for the cell to divide into two separate cells. The helix of the DNA has to unwind itself at the rate of some hundreds of turns per second, at the speed at which an internal combustion engine would disintegrate and fall apart. The new chain has to construct itself at the rate of some thousands of nucleotides per second, and then the process is complete. RNA is very similar to DNA, however with two unique differences. It consists of sugar ribose and also has a uracil base. As with DNA, RNA has four different types of codons, which are represented by the letters A, C, G and U. RNA also has 64 possible combinations of these types of codons. As DNA is unable to pass through the nucleus of the cell, it has to create a copy of itself, which is known as messenger RNA. If the DNA codes reads ATA, for example, messenger RNA will read UAU. There are three forms of RNA present within the cell. The first is identified as a carrier of the blueprint copy of DNA from the nucleus to the cytoplasm of the cell. The second is identified as ribosomes, which are present in the cytoplasm of the cell. And the third is identified as smaller molecules which appear when a ribosome bonds with a codon from the messenger RNA. The messenger RNA, which contains a copy of the DNA, passes into the cytoplasm, where a ribosome joins to its first codon and reads the code stored in the codon. This causes transfer RNA molecules to appear and translate the genetic code into a sequence of amino acids. The ribosomes are attached to the endoplasmic reticulum of the cell. Ribosomes serve as anchorage points within the cytoplasm for both the messenger RNA and transfer RNA, which, as mentioned before, are smaller than the messenger and ribosomal RNA. Each transfer RNA molecule bonds with a particular amino acid. There are three chemical groups known as the anticodon at one end of a transfer RNA molecule. They work in the same way that the base pairs in a DNA molecule do. There is a different transfer RNA molecule for each individual RNA codon. Now we come to the movement of code transfer, and for clarification, the sequence of code transfer from types of RNA are numbered. When number 1 messenger RNA joins to the ribosome, number 1 transfer RNA molecule carrying its amino acid bonds with the messenger RNA number 1 codon. The messenger RNA then moves one step forward in the ribosome, and number 2 transfer RNA molecule carrying the relevant amino acid bonds with the messenger RNA number 2 codon. The amino acid from the number 1 transfer RNA molecule connects with the amino acid from the number 2 transfer RNA molecule, which then leaves the number 1 transfer molecule free to find another amino acid. The messenger RNA then moves another step forward and another transfer RNA molecule arrives, and it receives the developing amino acid chain from the number 2 transfer RNA molecule. This process continues from hundreds for up to thousands of times until the chain reaches the full stop codon. When the number 1 ribosome has read the code in each codon, some length along the messenger RNA molecule, a second ribosome joins to the beginning of the messenger RNA and starts to make a second protein molecule, repeating the process. Later, a third ribosome joins to the beginning of the messenger RNA molecule, and then a fourth, fifth, sixth, and seventh ribosome joins, and so on. These different combinations of ribosome molecules being joined to the same messenger RNA molecule, each producing their individual protein molecules, are identified as polysomes or polyribosomes. They are in fact the protein factories of the cell. Now we come to the genetic code. All this information in the cell is consolidated in the form of a code of instructions, of which the 4 nitrogen DNA bases A, C, G and T are arranged as codons, which contain 3 letters in each one. As mentioned before, there are 64 possible combinations of these 4 letters of the code, which are in groups of 3. These combinations specify approximately 20 different amino acids that are present in living organisms. Some codons specify the same type of amino acid, whereas other codons serve as a full stop ending a particular protein chain. Put simply, the genes are set out in sentence form. Several genes combined become a paragraph. A large number of gene paragraphs or groups become a chapter, and a large number of chapters become a book, which is identified as a chromosome. It takes 23 chromosome books to create the complete encyclopedia of, for example, a human being. All this is then duplicated to create an exact identical copy, so that the human body contains 2 encyclopedias in each human cell. So then, there are 46 chromosome books in each human cell, and according to scientists, one DNA molecule actually contains so much information that it is like a library and would fill some 4,000 books of 500 pages in each. It is truly amazing that these books contain such a multitude of instructions, and that the human encyclopedias in every human cell are more than 35 times the size of our complete Encyclopedia Britannica. A truly awesome fact. Now, every cell that makes up a human being contains precisely the same genetic code. However, upon examination we find that groups of cells perform different functions throughout the human body. Some develop as kidneys, some as bone or muscle, nerve or lung, or the heart or brain, etc. Some govern the amount of melanin present in the epidermis and epidermal skin layers, which affects the colour of the human skin. Some govern the thickness of our bones, the colour of our eyes and hair. Some govern the shape of the skull, the length of fingers, arms, legs, and eventual height of the skeleton of the human body, etc. All the variations that are found in mankind around the globe are in the genes that are contained in every cell of our bodies as individuals, regardless of our so-called race and geographical location. What happens is that the DNA instructs each cell to perform a specific function. Each type of cell is governed by different genes from within the genetic code, which means that not all the genes present within the cell are actually used. In fact, only a few are. To describe this process, we have to look at the action of the proteins formed within the cell. The mechanism that causes one of a pair of genes to turn on or off works by the instructions of the dominant gene overriding the instructions of the recessive gene. Particular proteins known as repressors can inhibit or prevent the action of a gene by following the instructions of DNA and binding themselves to particular places in the DNA chain. This prevents messenger RNA from functioning at that place, and the gene is unable to develop into a protein molecule. Only a special molecule can remove a repressor protein by bonding with it. The genes would then function and translate normally. When a particular protein product has reached the correct level, the repressor protein bonds itself back onto the DNA. Thus, every component within the cell is designed to function in a specific, precise, and very efficient manner. The complete process of causing genes to turn on and off is governed by this extremely complicated and complex mechanism, which in turn is run by a fuel called mitochondrion, which provides the energy to fuel every organelle in the cell. Whereas all the components of the cell are governed by the DNA helix inside the nucleus, the mitochondrion fuel has its own instructions given to it independently by mitochondrial DNA, which is cited in the cytoplasm. Mitochondrion DNA has to run on the same fuel that it produces. Another fascinating scientific fact. The descriptions on the order and structure of our genetic makeup, as I have described, give some idea of the enormous complexities of the human cell. The functions of our genes are highly complicated and are absolutely essential for our existence from conception. In other words, the genetic makeup of each human cell has to be complete and functioning from conception. Otherwise there could be no human life. This scientific principle also applies to animal and plant cells and microscopic organisms. Armed with this knowledge, and with the fact that the laws of science are behind the whole function of the genetic code, it doesn't take a great deal of intelligence to work out that all this information, and indeed the DNA functions of every form of life, could only have come into being through the first cause, God Himself, the Creator of all things. The Bible states this in Romans chapter 1 verse 20. For since the creation of the world, God's invisible qualities, His eternal power and divine nature have been clearly seen, being understood from what has been made, so that men are without excuse. The discovery of DNA, RNA and the genetic code merely endorses that what the Bible says is true. So there is just one last thing to add here, and that is that true science is always a faithful witness to the truth of the Bible. Well, that's it for now. Thanks for listening. May the Lord bless you.